Welcome to Module 2 of Foundation Microbiology for Industrial Careers. In this module, we will classify the main groups of microorganisms and connect their biology to the risks they create in pharmaceutical, food, cosmetic, and biotechnology environments. Microbiology touches many industries because microorganisms can influence product safety, quality, shelf life, and regulatory compliance. Knowing which group is present helps us predict likely contamination sources, growth conditions, and effective control strategies. By the end of this module, you should be able to recognize the major microbial groups and distinguish prokaryotic from eukaryotic cells. You will also be able to relate microbial structure and survival features to quality control decisions in real industrial settings. Classification matters because a microbiologist does more than observe contamination. They ask what type of organism it is, and what that means. The answer guides the investigation of source, growth conditions, product risk, detection method, and preventive action. The major groups covered in this module are bacteria, yeasts, molds, viruses, protozoa, algae, and bacterial spores. Each group has distinct cell organization, growth behavior, and industrial significance. A fundamental distinction in microbiology is the difference between prokaryotic and eukaryotic cells. Bacteria are prokaryotes without a true nucleus, while yeasts, molds, protozoa, and algae are eukaryotes with a nucleus and membrane-bound organelles. Bacteria are single-celled prokaryotes and are among the most frequently tested organisms in industrial microbiology. They can be harmless, useful, objectionable, or pathogenic, and their morphology and growth characteristics help guide testing and investigation. Yeasts are unicellular fungi that commonly reproduce by budding. Although they are valuable in fermentation, spoilage yeasts may create gas, turbidity, odor changes, or visible growth in food, beverage, and cosmetic products. Molds are multicellular fungi made of branching hyphae and reproductive spores. Because their spores can spread through air and settle on surfaces, molds are important in environmental monitoring, raw material control, warehouse investigations, and spoilage prevention. Viruses, protozoa, and algae are not always the main focus of routine quality control testing, but they remain important conceptually. Viruses require living host cells to replicate. Protozoa are often linked to water or environmental systems, and algae are mainly associated with aquatic environments. Bacterial spores are dormant survival structures formed by some bacterial genera, including Bacillus and Clostridium. Their resistance to heat, dryness, and other stresses explains why contamination may persist, recur, or reappear after an apparently successful cleaning activity. Indicator organisms can reveal hygiene weaknesses, water system issues, handling failures, or broader contamination pressure. Objectionable organisms are specifically undesirable because their presence may create unacceptable product, patient, consumer, or process risk. Different industries prioritize different microorganisms because product composition, water activity, manufacturing conditions, and intended use are not the same. In pharmaceuticals, food, cosmetics, and environmental monitoring, classification supports a risk-based interpretation of microbiological results. This comparison table provides a quick way to distinguish the major groups by cell type, typical form, and industrial relevance. It reinforces that bacteria, fungi, viruses, protozoa, algae, and bacterial spores must not be approached with a single testing or control strategy. The class activity encourages you to compare one bacterial group, one fungal group, and one additional microbial group. Use cell type, shape, growth behavior, and industrial relevance to explain which organisms may be most important in pharmaceutical, food, or cosmetic settings. Gram-positive and gram-negative bacteria differ in their cell wall structure and gram-stain appearance. This distinction is not final identification, but it provides an important early clue for selecting the next investigation step and interpreting probable contamination routes. Bacterial morphology can provide useful preliminary information before advanced identification methods are used. Cocci, bacilli, spiral forms, and curved rods may suggest likely organism groups, but morphology must be combined with staining, culture characteristics, and confirmatory testing. Not all microorganisms are contaminants. Many play essential roles in fermentation, biotechnology, research, and quality control testing. The same microbial group may be beneficial in one process and unacceptable in another, depending on the product and manufacturing context. Industrial microbiology evaluates microorganisms by their effect on product quality, safety, stability, and compliance. Pathogens, spoilage organisms, objectionable organisms, and indicator organisms each require a different level of concern and response. When an isolate is detected, the first questions are what group it belongs to, where it may have come from, and whether it can survive or grow in the product. Those answers support a practical quality control decision, from trend monitoring to confirmation testing, deviation investigation, or corrective and preventive action. Product-specific risk depends strongly on formulation and use. Water-containing products such as oral syrups, lotions, and creams often require strong attention to bacteria, yeasts, molds, water quality, hygiene, and preservative effectiveness, while dry powders may be more vulnerable to spores and environmental molds. Classification supports testing strategy by helping the laboratory choose the sample approach, target group, culture media, incubation conditions, enrichment steps, and identification method. For example, Pseudomonas like growth in a water-containing cosmetic should trigger review of the water system, wet equipment, preservation system, and user-related product risk. In this case study, a water-based lotion shows cloudy growth, an odor change, and suspected gram-negative rods. A scientifically sound investigation should examine the water system, equipment hygiene, preservative performance, previous trends, relevant batch records, and appropriate corrective and preventive actions. This review exercise tests your ability to match clues with the most likely microbial group. A single-celled prokaryote indicates bacteria, budding cells indicate yeast, hyphae and spores indicate mold, a resistant dormant form indicates a bacterial spore, host dependence indicates a virus, and photosynthesis in water suggests algae. To summarize, microbial classification helps predict behavior, contamination source, product risk, and the most appropriate control strategy. As we move to the next module, we will build on this foundation by examining microbial cell structure, growth conditions, and their impact on industrial microbiology.